Hello, welcome to the CADASIL Connection webinar series. I'm Bert Kosiski, the president of cureCADASIL, and on behalf of myself and Vinita Ball, who is also a trustee of our organization, we would like to welcome you to what I think will be a very interesting webinar broadcast today. We're fortunate to have as our speaker, Elena Muiño, and she will be introduced by Chris Hopkins.
Okay.
Just a minute here. I'm going to try to advance the slide. There. cureCADASIL is a nonprofit organization dedicated to helping patients, families, caregivers, and other supporters touched by CADASIL. We aim to unite patients and the medical community toward the common goal of treatments and ultimately a cure for this rare genetic disease by promoting communication, advocacy, research, and education. Dr. Muiño is a practicing clinical neurologist and expert in bioinformatics. She gained experience in CADASIL while in Paris with the team that identified Notch3 as the cause of CADASIL. She received her PhD in medicine from the University of Barcelona. She worked as a postdoctoral researcher in the laboratory of Dr. Fernandez Cardenas, who is a recipient of the 2022 $1 million bike ride grant for cureCADASIL, and is at the St. Paul Institute for Biomedical Research in Barcelona, Spain. Single-cell RNA sequencing is a relatively new, sophisticated technique that Dr. Muiño and co-workers have been using to discover possible mechanisms causing clinical manifestations of CADASIL. They are using this technique in single cells obtained from skin biopsies from patients with CADASIL. This approach can be used to help discover not only mechanisms, but also possible therapeutic targets that may be mitigated or blocked by medications. Before we ask Dr. Muiño to present some of the results of her work, we have asked Dr. Chris Hopkins to give a brief introduction to the technique of RNA sequencing and how it is being used to understand the causes and possible treatments of genetic diseases. Dr. Hopkins is a Chief Scientific Officer of In Vivo Biosystems and a recognized expert in the field of discovery of genetic diseases. He has pioneered the use of models to study the effects of genetic mutations, like CADASILs, on living organisms. And he will give us some background on the use of RNA sequencing to help better understand Dr. Muiño's work. So I think we should.
All right, it looks like I've activated my slideshow. Can everybody see that now?
Mm-hmm.
Okay, great. Well, it's a pleasure to introduce Elena. And I'd like to thank cureCADASIL for allowing me to be one of the scientific board members. It's a joy to try and work with this team and figure out different ways that we can explore discovery in the Notch3 gene to see how variants in Notch3 lead to disease. One of the important things that Elena is going to tell you about is RNA-seq or transcriptomics. And how to understand transcriptomics, let's go look at sort of what we call as the central dogma in biology. And that is, how do we go from the information in our genes to how does that create all the proteins that we have that create everything that is us, creates all of our skin, our hair, and makes us who we are. Well, it's the central dogma, and that goes from the DNA that encodes the 20,000 genes. This then gets transcribed through transcription into mRNA. mRNA is the messenger RNA. It's a really important tool of the genes to to instruct the translational machinery that translates the mRNA message into protein. So that this is, by measuring mRNA, you're able to understand which genes are getting turned on and which genes are getting turned down, and that mRNA changes in level then directly translate to changes in protein. So mRNA transcriptomics is a proxy measure or an indicator for changes in levels of protein expression. So we'll be looking at mRNA as that tool, and there's a variety of techniques that have been used in the past. There was chips arrays, but the new techniques are using the NGS methods, where we can look at sequence reads, and this is RNA-seq, and that has turned out to become a very powerful method for identifying the biomarkers that can cause diseases. All right, so there's two different sort of utilities in RNA-seq. One of them can be done in bulk. That's where we take the entire tissue. Here I show you sort of a little skin biopsy. You can take the entire tissue, grind it up, and look at all of the RNA that comes from all the different cells, and that's a bulk mRNA harvest. This can be still quite useful, and is in fact generally how a biopsy might be used for in looking at a particular change in gene expression. Now, much more powerful technique has been recently advanced, and that is to take the tissue, separate it into single cells, and then look at each the changes in expression of genes in each individual cell. And that allows us to look at things like, so if we're looking at here, we've got your hair follicles and we've got the vasculature. We're really interested in the vascular changes in the vasculature. So we can harvest the cells coming from the vasculature and harvest the individual single cells and look at changes in gene expression in just the different cells that are involved in the vasculature. So it becomes a very powerful method for understanding disease biology at the cellular level. And with that, I would like to hand over the rest of the presentation. Hopefully that was a good introduction for us on RNA-seq, and let Elena take over and tell you about her exciting work in single cell sequencing to understand Notch3 variations.
So I want to interrupt just real quickly and tell everyone that all the audience is on mute. But if you have questions, type them in the question box that you should see in your panel. And at the end, we will do a question and answer session. So Elena, take it away.
I can share my screen. Okay. So, thank you for your nice presentation. Good morning, everyone. As already explained, I'm going to present our work called Single-cell RNA sequencing for CADASIL understanding and therapeutic target discovery. CADASIL is a systemic artery disease that affects all the small blood vessels in the body, although the symptoms are due to the central nervous system involvement. Blood vessels are made up of three main layers: the tunica intima, where the endothelial cells are found, the tunica media, where the vascular smooth muscle cells are found, and the tunica externa. In CADASIL, the cells that are mainly affected are endothelial cells and vascular smooth muscle cells. Thanks to the systemic nature of the disease, CADASIL could be diagnosed through the study of patients' skin biopsies, since the same alterations have been found in the blood vessel of the skin as in those of the brain. This is why the skin represents a very interesting tissue for the study of this pathology. On the other hand, CADASIL is due to mutations in the Notch3 gene with an autosomal dominant dominant inheritance pattern. This means that each child of a CADASIL patient has a 50% chance of having the disease. The Notch3 gene gives rise to a protein also called Notch3, which is a cellular receptor that transmits signals from other cells. It is involved in the formation of blood vessels and is mainly expressed in the vascular smooth muscle cells. Hence, the importance of the study of this cell type, since it is the main cell type together with endothelial cells that are affected in the disease. The Notch3 receptor consists of an intracellular, a transmembrane, and an extracellular domain. The latter contains a region with 34 repeated amino acid sequences called epidermal growth factor-like repetitions. Mutations occurring in CADASIL result in an odd number of cysteines in this particular region, leading to misfolding of the receptor and aggregation of multitudes of proteins. But the mechanism by which this leads to the disease and symptoms is not well established. It has been hypothesized that this protein aggregation may sequester proteins or cause them to gain a toxic fashion, as in the case of TIM3 or big-tronectin, giving importance to the role that the extracellular matrix may play, as the proteins affected are immersed in it. CADASIL is a rare disease, as it has been estimated to affect less than 1 in 2,000 people. However, from the study conducted by Julie Rutten and collaborators, they realized that 3 in 1,000 people have the characteristic mutations of CADASIL. With this and other subsequent studies analyzing databases such as the UK Biobank or Genome ID, they realized that patients who came to the doctor's office have mutation affecting epidermal growth factor-like repetitions 1 to 6, and individuals from biobanks without a specific disease have mutations affecting epidermal growth factor-like 7 to 34, leading to the hypothesis that the location of the mutation may be a determining factor in the prognosis of the patients with Notch3 mutations. 4 are the cardinal symptoms of the disease: migraine, psychiatric disturbance, recurrent ischemic strokes, and dementia. Characteristically, the cognitive domains mainly altered in CADASIL are executive function, ability to make plans, as well as attention and speed of information processing. The suspicion of the disease is mainly due to the characteristic MRI lesions in which we find white matter hyperintensities affecting mainly the external capsule and the temporal poles. The diagnosis can be made through the study of the skin biopsies, but currently the gold standard is the genetic test. The prognosis of patients with CADASIL is unknown. As mentioned before, it seems that the location of the mutation may have an influence, and it also seems that males, the number of small subcortical infarcts, and cerebral atrophy are associated with a worse prognosis. There is no specific treatment for the disease, only for cardiovascular risk factors, and there are novel and advanced therapies that are in development, but are still in the experimental phases with animal models. However, the possibility of drug repurposing, in which drugs already approved and marketed for other diseases may have a new use, has never been explored in CADASIL. For example, galantamine was initially used as a drug for poliomyelitis and is currently prescribed for patients with Alzheimer's disease. Therefore, we need therapeutic targets. Omic techniques allow the study of a large number of molecules to understand the etiopathogenesis of diseases find associations with characteristic phenotypes, establish causality, identify biomarkers, or find therapeutic targets. Our recent review with respect to the advances that omics technology had brought about in CADASIL pointed to several molecules as relevant targets for the disease, as well as several pathways like autophagy, cell adhesion, mitochondria, or the TGF-beta signaling. We have already carried out a genome-wide transcriptome study in skin biopsies of CADASIL patients, where we performed a punch skin biopsy, and the whole sample was homogenized to later extract the transcripts and analyze them. We found that higher levels of E2F4 mRNA in CADASIL skin biopsies compared with controls, a transcription factor associated to endothelial cells migration, neuronal survival in ischemic situations, and TGF-beta pathway. E2F4 highest levels of expression were associated with the worst executive function and attention and information processing speed, the two cognitive domains characteristically most affected in patients with CADASIL. Single-cell RNA sequencing allows transcriptomic studies by cell type. A skin biopsy is preprocessed to dissociate the tissue and isolate the cells. Subsequently, each cell is marked so that when each transcript is sequenced, we know which cell it belongs to. As each cell type has more or less specific transcript, a correlation is made between the sequence molecules of each cell and the known signature of different cell types to identify which cell type we are dealing with, and to be able to select specific types in subsequent analysis. Therefore, we are facing with a rare disease, probably underdiagnosed due to mutations in NOTCH3, a gene that is mainly expressed in vascular smooth muscle cells, cells characteristically affected in the disease along with endothelial cells, with an unknown prognosis and no treatment. Understanding the disease through transcriptomic study of those cells characteristically affected in CADASIL will facilitate the identification of molecules relevant to CADASIL that can be used as therapeutic targets in cellular models of the disease for subsequent clinical trials of drug repurposing. So our aim was to identify transcripts associated with CADASIL severity to find potential therapeutic targets and biomarkers. So subjects were selected from Cadagenia. This is a registry in which patients with mutations in NOTCH3 have been consecutively recorded since 2017 from different parts of Spain, mostly from Catalonia. Epidemiological data, cognition, and neuroimaging profiles and skin biopsies were registered. For this study, the cohort was balanced by sex and mutation location. Likewise, patients were of similar age to avoid bias. For this study, updated clinical information was collected, as well as extensive neuropsychological tests. To assess the severity of the patients with CADASIL, we use an ordinal scale of cognitive impairment according to normal, very mild, moderate, or severe impairment. We also evaluate associations with the domains typically affected by CADASIL, the executive function, and attention and speed of information processing. As secondary outcome, we wanted to find transcripts associated with the different position of the mutation. Executive function was assessed by means of verbal fluencies, the Stroop color word test, and the trail making test part B. and attention and information processing speed was assessed through the symbol digital modalities test, the Stroop word and color test, and the trend making test part A. Raw scores were adjusted into CETA scores by age and years of education, following Spanish normative data. A higher adjusted CETA score indicated a better performance in all cases. And we calculated cognitive domains indices by averaging the adjusted scores within each domain. 2 4mm skin punch biopsy were obtained for each patient to achieve a large amount of tissue, avoiding the need of stitches. To obtain the cell suspension, a manual setup was carried out and compared with the use of a commercial kit to study which of these pre-processing methods obtain better cell viability. For both pre-processing methods, we use CD31 labeling and endothelial cell-specific labeling to ensure that vascular cells, which are involved in the etiopathogenesis of the disease, were obtained with these procedures. After dissociation, the samples were taken to the Josep Carreras Leukemia Research Institute for library preparation and subsequent sequencing. Briefly, about 12,000 cells were loaded in a well of the Chromium Next Gene Chip G from Tenex. The Chromium controller performed single-cell partitioning and barcoding. The cells were partitioned into nanoliter scale gel bits in emulsion genes were all generated cDNA will share a common Tenex barcode. Tenex barcodes were used to associate individual reads back to the individual partitions. A series of quality controls are necessary for data analysis. For this, we use R version 4.3.1 and the Zebra toolkit. Initially, for each patient sample, low quality or dead cells were removed. This is given by the percentage of reads that map to the mitochondrial genome. Also were removed anti-droplets, doublets, or multiplets, where the number of unique genes detected in each cell, as well as the total number of molecules detected with a cell, are too few or too many. After removing unwanted cells, a transformation of the data is carried out to normalize the expression information. Once all these steps have been completed, we can integrate information for each patient. Subsequently, the data will be scaled so that the mean expression across cells is 0 and the variance is 1. This step gives equal weight in downstream analysis so that highly expressed genes do not dominate and bias our results. Once at this point, we proceed to perform a linear dimensional reduction to a principal component analysis, and then a non-linear dimensional reduction through the uniform manifold approximation and projection. It will now be possible to find clusters and know to which cell type they correspond according to the expression of the different cell markers. Finally, we can perform a differential expression analysis with Zebra package. In our case, according to the severity of the patients and according to the location of the mutation. These are the main characteristics of the study patients. The age range is between 49 and 61 years old. 50% were women. In 50%, the mutation was located in the epidermal growth factor like repetitions 1 to 6, and the 50% had mild impairment. The rest of the patients had at very mild deteriorations or no deterioration at all. But we cannot have any patients with moderate or severe cognitive impairment. We conducted two kinds of studies, one evaluating patients with very mild or normal cognitive impairment versus those with mild cognitive impairment, and another according to the location of the mutation. Interestingly, in our cohort, patients with mutation located in the exons coding for the epidermal growth factor-like receptor repetitions 1 to 6, where the cognition was better. This was probably related to their younger age, since patients with epidermal growth factor-like 7 to 34 involvement tend to come to the clinic older. For the choice of the preprocessing protocol, the following were considered: several enzyme concentrations, buffer temperatures, speed and type of agitation, cell counting method, sample storage, and finally, both preprocessings, manual and with the commercial kit, obtain similar viabilities, cell viabilities. So the later one was chosen for its simplicity, the kit. These are the violin plots of the quality controls for each of our 8 patients. For each is represented the number of features, which reflects the number of genes with at least one transcript, the number of counts, which is the total number of molecules detected in each cell, and the percentage of reads that map to the mitochondrial genome, as low quality or dying cells often exhibit extensive mitochondrial contamination. Here we perform the UMAP in order to distinguish clusters corresponding to different cell types. If we color by patient, we can see that these have been distributed by most of the clusters, indicating that the integration of the data has been correct, since we do not see that the cluster represents patients. This is a cluster with several patients, but not just one patient. And instead, they represent different cell types. And here we colored the endothelium in red and the vascular smooth muscle cells in blue. The total of patients evaluated were 8, with about 12,000 cells that passed the quality control and about 30,000 different genes. 1% of the cells were endothelial and 7% were smooth muscle cells. In this volcano plot, we represent the analysis according to the cognitive impairment presented by the patient regarding the cell type study. The orange dots represent significant transcripts after adjusting for multiple comparisons, and the lilac dots represents those that were significant, and the absolute value of the log2 fold change was greater than 2. These transcripts are expressed in patients with mild cognitive impairment versus those with no or very mild impairment. This graph shows the relationships between the transcript found significant in our study according to cognitive impairment in the vascular smooth muscle cells. We can see that almost all of them are related in some way, either by experimental studies or by text mimicry. From the biological process of gene ontology, there is an overrepresentation of terms related to apoptosis, mitochondria, and energy metabolism and vascular development. From the molecular function of gene ontology, there is an overrepresentation of terms related to energy metabolism and cell adhesion. And one of the significant HEEGG pathways was Alzheimer's disease. In this volcano plot, we represent the analysis according to the mutation location of the patient regarding the cell type studied. The orange dots again represent significant transcripts after adjusting for multiple comparisons. The lilac dots represent those that were significant and the absolute value of the log2 fold change was greater than 2. These transcripts are underexpressed in patients with mutations that affect epidermal growth factor-like repetition 7 to 34 compared with those with affecting epidermal growth factor-like repetitions 1 to 6. Again, we can see that almost all the transcripts associated to the mutation location in the vascular smooth muscle cells are related in some way. From the biological process of gene ontology, there is an overrepresentation of terms related to apoptosis, cell differentiation, extracellular matrix, vascular development, or ubiquitination. From the molecular function of gene ontology, there is an overrepresentation of terms related to ubiquitination, extracellular matrix, TGF-beta signaling, and cell adhesions. So these are the 5 transcripts found to be significant after adjusting for multiple comparisons and with a log2 fold change in absolute value greater than 2. The study in endothelial cells, according to cognitive impairment and mutation location, point to FABP4. This molecule has been related to the TGF-beta pathway, endoplasmic reticulum stress, and autophagy. Processes studied in CADASIL and found to be altered. RPS4Y1 and RPS26 are ribosomal proteins that have been associated with dementia. In our study, according to cognitive impairment, RPS4Y1 was the only one that was found to be significant and with an absolute log2 fold change greater than 2. This molecule is also related to cellular adhesions, therefore of interest in CADASIL, where it has been seen in many histopathological studies how these are affected. SERF2 is a protein related to the assembly of amyloid fibers, a substance related to amyloid angiopathy and Alzheimer's disease. It also promotes protein misfolding. This is of interest in a disease due to misfolding of NOTCH3. And for lastly, IGF-BP5 that was also related to dementia and TGF-beta signaling. The study of gene ontology terms and pathways show us that there are several functions of interest in the study of vascular smooth muscle cells, with a large overlap between the study according to cognitive impairment or according to the location of the mutation. They are apoptosis, cell adhesions, cell differentiation, vascular development, mitochondrial energy metabolism, Alzheimer's disease pathway, ubiquitination, extracellular matrix, and TGF-beta signaling. And all of them have been at some point directly or indirectly related to CADASIL. Regarding the challenges of the study, we found a low sample size as is usual in rare diseases. However, it is overcome by the fact of studying hundreds of cells. We do not have a replication cohort, but in the next year, we expect to have new data to confirm our findings. The delay that did not allow the replication to be carried out at the time of the study was mainly due to the great technical complexity of the preprocessing of the samples and my maternity leave. However, we believe that despite these challenges, we have managed to overcome them and carry out a study of great interest that provides both molecules and pathways as potential therapeutic targets. As next steps, we will complete the differential expression study according to the scores on the scales assessing executive function and attention and speed of information processing. We will perform a new single cell study with new patients to try to validate the results obtained. In parallel, we will perform an inhibition overexpression of those genes with adjusted significant p-values and log to fold change greater than 2 in cellular models of the disease, as well as an inhibition or overexpression of pathways such as autophagy in order to improve the cellular model and subsequently test it in animal models. So as conclusions, single-cell RNA sequencing of skin biopsies from CADASIL patients can be performed by manual or commercial kit pre-processing with comparable cell viabilities. The study in endothelial cells and vascular smooth muscle cells has revealed potential therapeutic targets in CADASIL, FABP4, RPS4Y1, RPS26, SERF2, and IGFBP5. The study in endothelial cells and vascular smooth muscle cells have revealed potential functions and pathways to be targeted in CADASIL as autophagy, cell adhesions, ubiquitination, extracellular matrix, or TGF-beta signaling. Both the transcript and associated pathways have functions described in the literature as adherent in CADASIL, which supports our results. Thank you for your attention. Thank you very much to the group of stroke pharmacogenomics and genetics, and to all family members, caregivers, or patients with CADASIL. Thanks to the CADASIL Spain Association for their support, cureCADASIL, and the $1 Million Bike Ride program for their trust in us. Without you, none of this would be possible.
So thank you very much, Elena. If people have questions, and if you can type them in the question box or in the chat, we'll find them there. Okay, there is a question in the chat, and the question is, how do you go about testing the potential therapeutic targets?
Well, for about the potential therapeutic targets, first of all, we will try to replicate our results to make sure that really a possibility. But also in parallel, we are going to try to prove if the inhibition or overexpression of this transcript could change the phenotype of the cellular model. The cellular model, thanks to the reprogrammed mature cells to stem cells and then to mature cells that is interested for the disease, could aid us in this purpose. Now, our partners in Santiago de Compostela has a cellular model with the immature cells, but in these immature cells, they can already see differences between CADASIL patients and control. So we are trying to perform, I must say, to modify the autophagy process to see if there is an improvement in Notch3 aggregation, because in CADASIL, the typical aggregation is extracellular and the most studied, but also there is a intracellular aggregation. So we are trying to see if modifying autophagy, we could change the pattern of aggregation. But this is the beginning, and we think we are in the right way, path. I don't know if this
So
is the answer. I cannot see the questions. How can I see the chat, or I cannot?
well, one of the questions was in the chat, and I'm watching for questions in the question and answer box.
Okay,
A more general question, just about using the technique, and looking for the transcriptomics in general. It's probably not enough. I mean, in terms of finding a potential surrogate marker or endpoint, isn't it more like hypothesis generating, and then in the end they'll have to go back and actually see the protein or other measurement that will confirm it before it comes into a clinical realm, for example, endpoint in a trial?
Yes, I share the opinion of this individual subject. There is not the only solution. I think it's just one point. You have to see the whole picture. Only the transcript is not the only thing. Because of that, we also are working proteomics to try to then integrate all the information. The only point is the proteomic study is in blood samples instead of the skin biopsies, but we think that we can have more information with proteins. But anyway, I think transcript could add. And if we replicate our analysis, they point us to several pathways that is in the literature really already mentioned. So I think the results are robust from that point of view. And because of that, to try to inhibit and also overexpress the same transcript, even not knowing what happens to the protein, could aid to have more information in the cellular model, and also the information we will have about proteomic study could be really interesting.
Did you see any evidence for a signal for inflammatory responses? Because I think I've seen that elsewhere in the literature.
Yes, there could be a role of inflammation, but we, in these results that are preliminary results because of the delay, we have the last analysis last week. We have not seen specific or a lot of inflammation pathways or molecules related with this. There are some pathways or GO terms in the analysis performed with a string, but not since the most relevant path. But it's something that we want to explore.
Great. I'm going to change here and show.
I can see. I can see one question. That, can you speak to why E2F4 from bulk was not seen in single cell analysis? Well, I'm not digging a lot about this topic, but the point is I didn't. It could be that there is not in the chip in the futures this E2F4 because there are about 30,000 futures, so not all molecules are. And in addition, could not pass some quality control of the sequencing machinery, and maybe because of that. Because I didn't found E2F4 in the futures.
Also, another question that I missed. And was there a difference in the exon 1 to 6 versus 7 to 34? Did you see any differences in transcription?
Yes, the difference between the location of the mutation.
Right.
Yes,
Whether it was in.
We saw Really similar results among those with different the analysis of cognitive impairment and the location mutation. And actually, the difference in the location mutation analysis was the one with more significant transcript adjusted for multiple comparisons, and also with a log to fold change greater than 2 in absolute terms. So.
Another question in the chat, do you have any plans to look at more severe patients for these same biomarkers?
If we are going to try to record severe patients, for sure, also for replicate the study, because a limitation now of our study is that all the patients were a lot of, they were good. They only have mild impairment. So we are going to recruit more patients with more severe phenotype to try to replicate our results, or to find more different results.
Another question, can you speak to why the E2F4 from the bulk was not seen in the single cell analysis?
Yeah, is the question before I answer, is because I think it was not in the final analysis. It was, the future was not in the analysis, because I checked it and it was not in the analysis. But I will check again and ask to the platform, to the next, if they do not have this transcript, or maybe it was lost in the quality control analysis of the sequencing, or whatever.
And then maybe finally here, how would you go about, and do you have any candidate repurposed drugs that you could test in to see if it has an influence on the transcription?
I have not already tested, I have not already searched which drugs could modify my targets, or our targets, but normally there are platforms, softwares that we are going to use to try to find targets. Not only for my specific molecules, but also for the whole analysis, taking care about the weights, the p-values, etc. So it's the next step also.
Okay, I think we'll probably end it there. Thank you very, very much for the presentation. It was very thought-provoking and interesting. For everyone, the recording of this will be available. It usually takes a couple of days, but I think by the end of the week, the recording should be available. We will post it on our cureCADASIL website, and so people can go and look at it again and review things if you didn't catch it first time around. So again, thank you very much, and we hope that we'll see you all in our next webinar.
Hope so.
Okay.